Hello everyone, and welcome to our lab exploring the diversity of plants in the greenhouse. For the first assignment is to start this lab off on the right foot. We want to make sure that you have a recap of the material from the lecture, meaning that you're able to draw the basic structure of the tree of life, so that way it actually means something to you when we go take a look at a bryophyte versus a fern versus a gymnosperm. So as we have a tendency to do, let's draw up the tree of life. We of course know that life evolved from an aquatic, anoxic environment, no oxygen. The first diverging group was that of the bacteria. I'll move through this quickly since hopefully this is all second nature to you by this point. We of course have the archaebacteria, and then we have the diversity of the eukaryotes. Now, within the eukaryotes, we of course have that large polytomy of many different protist lineages, one of which are the archaeplastids. Or archaeplastida. What defines the archaeplastids was the endosymbiotic event of a cyanobacteria ancestor. That today is their chloroplasts, helping them perform photosynthesis. Now, this group diversified into a lot of different algae. From your lecture, hopefully you remember that the first diverging group of algae were the red algae. Different color, have multicellularity in a different form. After that, we have the diversity of our chlorophytes and our charophytes. Before we finally get to our land plant diversity. Now, I'd recommend you go back to your lecture and see if you could figure out some of the traits that differentiate those groups. We're going to focus instead on the actual plant tree of life for the purpose of this lab. The first group, collectively known as your bryophytes, are not down their own branch, but instead are a sequence of 3 independent branches on the plant tree of life. Those branches are that of the liverworts, the mosses, and the hornworts, collectively known as our bryophytes. Now, one of these traits that unifies all of our bryophytes are that all of these organisms are non-vascular. They don't have true veins. They, therefore, remain very, very short. You can also talk about the fact that they're haploid dominant or gametophyte dominant. Those are traits that we'll talk about once we get to looking at those species. After our bryophytes, we then have the differentiation between those lineages that have small leaves, single-veined leaves, our lycophytes, and everything with a megaphyll, leaves with multiple veins per leaf. Down our megaphyll-bearing branch, we have the ferns and their allies. Of course, hopefully you remember the several different groups that exist down that branch. We will be exploring the several different families in that group, the horsetails, the whisk ferns, and the true ferns today. And then of course we have the differentiation from everything that bears a seed. Because everything prior to this had spore-based reproduction. Those things that carry seeds are either seeds carried in a cone, like that of your gymnosperms, or your angiosperms, which differentiate into 2 major lineages, your dicots and your monocots. So that's a quick recap of the tree of life. I've left some of the traits on there, like the fact that seeds unify these different species. Your task, in order to get the lab started, is to fill in the rest of it with different traits. Once you're done with that, I'll see you over by our terrarium tanks to take a look at some of our bryophyte diversity. So now that we're refreshed on our tree of life, we can now take a look at the very base of that tree of life for plants, which is very fitting because that's exactly where you find most of your bryophytes. These non-vascular, photosynthetic, gametophyte dominant plants are quite cute and quite tiny. So here what we're looking at is one of these gametophytes. You'll see that there is some branching, but if you take this under a microscope, you won't see any veins. They're not truly differentiated cells. These are kind of all the same cells with slight repurposing. This is very similar to what you'll see in the animal tree of life, where the earliest diverging animals, your sponges, are one cell type repeated over and over with slight specializations in order to eat stuff. In this case, plants are not eating stuff. They are instead photosynthesizing, and in some cases, trying to anchor themselves into the soil or on the side of a tree. Now, what's very important about these mosses is that they have the alternation of generations where their green stage is their haploid gametophyte stage. So the entire body of this thing, the green stuff here, and there are no sporophytes, so this is all gametophyte, this is the equivalent of the egg or sperm body, because these cells will replicate up until they form a structure that can go on to make those specific egg or sperm cells. The same is true for this other moss here. It also is your gametophyte body, but you'll notice that there is at least one sporophyte forming from this cluster. It's still immature, but it will eventually dry out when the spores are ready. And it is this structure here that's actually diploid. You can see more of them if you take a look into our little terrarium. So we'll try to stick the camera in there. I'll grab the thermometer, see if I can point at them. So this is a little bit happier of a moss specimen, and you can see, nicely done, Becca. You can see the little sporophytes sticking out. There, and then back here. Those are the diploid life state for our mosses. Let's go back up to the board to make sure that we're able to really understand what's going on with this, because it's so unique and so weird, but so characteristic of our plants. So with the alternation of generations, you have two distinct stages. A sporophyte stage, meaning the spore-producing plant that is diploid, and then the gametophyte stage. That is your gamete-producing plant. These will truly make your egg and sperm, but they are the body of the egg and sperm. Both of these stages can perform mitosis. And when you can perform mitosis, we call you a generation. Hence, the alternation of generations. The sporophyte will go on to make spores via the process of meiosis. Those spores will land somewhere and replicate, make bodies. These, in mosses, are photosynthetic. In all of your bryophytes, the gametophytes are your photosynthetic dominant stage. The gametophyte will then go on to make either egg or sperm or both. And then when one egg and one sperm fuse in the process of fertilization, you then go on to make a sporophyte that will make those spores again. And so that's the repeated life cycle of these mosses. All of the green gathers the energy, makes the egg or sperm. When those fuse in whatever appendage that they are in, they then will temporarily form a sporophyte, that long stalk, that will create spores, but is nutritionally dependent on the gametophyte stage. Those spores will go ahead and shrink down via meiosis to make more of your gametophytes, and the process cycles. Now, of course, the reason I'm making a big deal of this is because the bryophytes are gametophyte dominant. Once we get past these and start looking at organisms plant organisms that have leaves, whether they be lycophytes, ferns, gymnosperms, or angiosperms, they're all sporophyte dominant. So take a minute to chew on that. Of course, this video does require a lot of pausing and drawing, so I'd encourage you to back up the video and draw some of those mosses and really pay attention to what the sporophyte looks like compared to the gametophyte. It's really telltale once you look at the branching structure of those appendages. Have fun with that. See y'all in a few.
So as promised here, you see the bryophyte alternation of generations, the gametophyte and sporophyte, so a little bit inverted to how I drew them, but obviously the order doesn't matter here. You still have the same separation with mitosis in the sporophyte interrupted by meiosis, mitosis in the gametophyte broken up by fusion or fertilization. And then we have a closer up image of the sporangia, the sporophytes of our bryophyte lineages. These being the diploid structures, and the green stuff being the haploid structures.
All right, so I'm back over by the terraria where we've got some of our club mosses, some of our lycophytes. You'll find a plethora of different Selaginella species. These are a member of the lycophyte family, but they've got a couple of unique features that distinguish them from true lycophytes. No lycophytes in the greenhouse or here for now. So let's take a look at this up close, because you're looking for those defining features of a microphyll. So hopefully you have a chance to handle this in person, but in order to see these, you should really be looking for those tiny individual leaves. And you're going to want to take that individual leaf and put it under a dissecting microscope to see the fact that it's got no vein, or a singular small vein to it, no branching. This is true for different species of Selaginellas that also have only a single vein. I like this species a lot because you can see the white indentation on these leaves where the vein is present. Now we've got a couple other Selaginellas in our terraria. So if you come over here, this species is thriving and you can see all of its tiny little microphylls, no spore producing structures. You can tell that this species here is not a Selaginella because of the fact that its leaves have many veins on the underside. This is indeed a fern. You've got another Selaginella species back here, characteristic with its branching pattern. So that's the other thing that you want to be paying attention to, the fact that this dichotomously branches, or branching in forks like a V. And then if you take a look into this tank, we again have a Selaginella over here in the corner. And then something I didn't highlight last time with our bryophytes, you've got a liverwort here, thriving, growing quite happily from this thallose body to now these bodies that are growing and expanding and going to first extend out with a long appendage and then kind of flesh out and flatten out to create that flat body. So next up, we're going to be talking about our fern allies. Spend some time drawing the microphylls, and I'll additionally have another photo of the microphyll under the dissecting scope, and then we'll reconvene when we're ready to draw our ferns. All right, so now we're in the greenhouse where some of our bigger megaphyll bearing lineages are. We've abandoned our lycophytes with their microphylls and our bryophytes with their non-vascular shortness. And now instead we're going to take a look at some of the fern diversity, fern and allies, because not everything in that group is a true fern. But what they all do is they all produce spores and they all have megaphylls. So let's take a look first at Equisetum. This, in this area, I've heard it called snake grass. I grew up calling it horsetail because it will have megaphylls that branch out from each of the nodes on the stem. Now, if you come in really close, you can see, I don't know how the focus is on this, you can see the little divots where the true leaf has been aborted, but its boot print is still there. And that's what you're seeing across each one of these nodes. The megaphyll doesn't need to grow anymore because in this particular species, the stem is sufficiently photosynthetic. But all of the equisetums, all of your horsetails, have this wraparound megaphyll, multiple veined leaf. Now you're going to see the fact that it is multiple veined when we take a look at this under the microscope when we head back in the lab. Now another fern ally, that's actually my favorite of the fern allies, has a reduced megaphyll. So this is Psilotum, the whisk fern, because guess what? This looks like you could whisk together something. Now the whisk fern is very, very unique because it is an example of a simplification after evolution. So in this case, it had megaphylls, but the megaphylls have shrunk down to the point now where you still have this dichotomous branching pattern. You still have spore-bearing structures called synangia, the yellow clusters on these leaves. But you don't really see leaves on this whole thing. You just see tiny little annations because it's just lost the genetic architecture to make megaphylls. And that happens a lot in evolution. Your ancestors had something, you've since lost it. Look no further than our tails for a similar story, right? So we've got Psilotum that we'll again take a look at under the microscope with its reduced megaphylls. And you've got Equisetum with its true still megaphylls. For our ferns, we'll have to look up to one of our major Boston ferns. Now, if I remove the booby traps, so that's going to come a little bit closer, you can take a look at the veination patterns in each of these fern leaves. Hopefully the sun is just right. Actually, Becca, if you come swing the camera around here with the sun in the backdrop, you can see the veins much, much better. How's that? Perfect. So you can see the many, many veins in these leaves. And now I'd encourage you to additionally take this under the microscope and see the branching pattern, because what you'll find is dichotomous branching in each of the little veins coming off of the main vein of the fern leaf. Now this fern, as a spore-bearing plant, will make what are called sori on the underside of the leaf. Now it's not the mating season for this Boston fern. Instead, it's oftentimes reproducing by sending out creeping rhizomes, these roots that will take a hold in another plant, and then all of a sudden a whole other plant will start developing from there. But you will see some of these leaves growing. When a fern leaf grows, it grows as a fiddlehead, a wrapped around structure with something called circinate vernation, because it's unfurling in a kind of unfolding way. Whereas when you have leaves of some of our angiosperm plants, rather than unfolding, they'll kind of just expand out, like this pothos here. So, pause the video as necessary. Make sure that you can draw the leaves of our ferns, our whisk ferns, and our horsetails. And when you're done with that, I'll see y'all for our final discussion of all of our seed-bearing plants.
Back to our lycophyte, the Selaginella, under the dissecting microscope, so that way we can hopefully see the mid-vein of the microphyll in more stark contrast. So here's what we're looking at, a single microphyll with a singular vein offshooting from the stem here. And this really is what defines the microphylls of lycophytes. One unbranched vein to be very thin, with leaves of corresponding small width. Now there are some lycophytes that have extremely long leaves, but none of them have wide leaves because of the rules of diffusion. You cannot get resources, you cannot get water very far with leaves that are not, or with veins that are not branched. I'd also like to point out the absolute behemoth of cells that these leaves are made of. You can see them under the dissecting microscope. And of course, we also have our two different ferns, or fern relatives rather, the Psilotum, whisk fern, and Equisetum, horsetail, underneath the compound microscope to show the differences in the stem architecture. You can see the singular vascular bundle, the one vein running down the middle of Psilotum with its reduced megaphyll habit. So rather than making leaves, it's really not making any. Small innations are the best it's got. You have one vascular bundle running up and down, and compare that to our giant equisetum stem, wherein you see lots of little vascular bundles that support the growth of megaphylls. So it's the distinction of these bundles and the lack of distinction that allows you to grow either megaphylls or no leaves, or at best microphylls. And this is actually similar to what the stem of Selaginella would look like. A singular vascular bundle, compounded in the middle, whereas megaphyll-bearing lineages have these more giant vascular bundles. Now, before getting y'all into the greenhouse for the discussion on our gymnosperms and angiosperms, I thought it kind of neat to take a look at some common crop plants growing here in the summertime in Michigan. So we have a dicot squash with its netted veined leaves and a monocot corn plant. You'll notice both of these have flowers. Here you see the male flower protruding for our corn plant, and here towards the center you have the female flowers of our squash plants. Yes, they do have different male and female flowers. Now for some other plants, one that's not native to the area, the Hoya, a plant in the dogbane family, as you'll notice this dog off in the distance, not coming near it. The family's named the dogbanes because some plants in this family have toxins for dogs. They can kill them and have been used historically as poisons for them. But you'll notice, as I point out in the video, the neat trends in terms of flower dynamics here, or flower structure here. And then, of course, you've got the differences here in the downy rattlesnake plantain. A plantain, meaning that it is a monocot. This is actually a member of the orchid family. Very, very vibrant leaves that look really cool. And then the real reason why I wanted to show you these plants was because I took some of those Hoya flowers underneath the dissecting microscope. And you can see the patterns of five. The five outer, kind of hairy, waxy structures, the five inner petals, and then even down to the male reproductive parts, the anthers, there are 5 of them here. So it just kind of matches this blueprint. Let's now head off to the greenhouse and look at some of our gymnosperms and angiosperms there.
All right, so we're back in our corner here, looking at some of our plant diversity, but this time I now have a giant pine cone. This is a sugar pine cone. And you're supposed to take a look at some of the individual ovuliferous scales. You'll see the fact that 2 ovules are carried on each of these scales because you see that pattern of discoloration that is not the size of the actual ovule and seed, but rather where the tissues originally developed when they were very, very small. And all of these scales carry that bootprint, even though the pine cone is absolutely huge, several, twice as big as my actual head. Now these gymnosperm plants that carry seeds in a cone, are oftentimes defined by having much tougher leaves, like this Norfolk Island pine. I know you can't experience the tactile experience via video, but it's a very tough, feels very unnutritious. We have another large specimen of our Norfolk Island pine here, and I'd encourage you all to feel the plasticky nature of these plants as they grew to defend themselves from the major herbivores on the planet when they were thriving, things like the dinosaurs. Now, of course, you might better recognize some of our small pine saplings as some of the species that, some of the gymnosperm species that dominate our area today. But in those pine needles, you'll notice that they cluster up together. They have multiple veins per pine needle. They still carry those megaphylls, and their reproductive features show us that they are no longer the spore-bearing plants like we saw in our ferns, but rather seed-bearing plants. And that leads us to our last major diverse group of seed-bearing plants. Caught that on camera, right? Our angiosperms. Here, we're fortunate enough today to have a flowering angiosperm, the Hoya plant, with its absolutely gorgeous flowers. Now from lecture, you'll hopefully be able to recognize the fact that these plants, with their 5 appendages, are dicots. The dicots typically have flowers in parts of 4 or 5, whereas our monocots have flowers in parts of 3s. When you think of flowers in parts of 3s, think of a vertical section of a banana. You can split the banana if you poke it from the top end or the bottom end. You can split it down the middle, down the length of the banana, into 3 individual parts. Those correspond to the 3 different chambers of the ovary. You'll see that 3 sections of fruit or flower alongside the parallel veins, like in this bamboo. Hopefully you can see the fact that all of the veins run parallel to one another, as this is one of the defining features of many of our monocot lineages. But not all of them. You do have some monocot lineages that have some patterns, some patterns that look like this, if they're some of our basal monocot lineages. And you can additionally see that parallel vein pattern innovated on in the parallel nature of this banana, our little dwarf banana plant. Still parallel veined, but not in the same way that the grasses are parallel veined. Now for your dicots, you can really explore the greenhouse and take a look at a lot of these different plants. The dicots are defined by having netted veins. So you see a wide diversity, a wide sprawl of veins in these dicot leaves. Or in these dicot leaves, which very fortunately have been nursed back to incredible health by Becca, the calicarpa, the beauty berry, a native to the southeast, Florida, that has these absolutely stunning pink berries. Its leaves are incredibly netted. You can also look at our beautiful coleus for examples of its absolutely monstrous, beautiful netted veins, as well as our croton, or our begonias. So have some fun. Utilize Wikipedia, explore the greenhouse, and as has been the nature of this entire lab, draw the patterns you see in plants, because it is from their anatomy that we can infer their location upon the phylogeny. And the entire purpose of this week's lab is to try to see if you can place different plants along the phylogenetic tree. That's what's going to be asked of you come final time. Build a character state matrix of some of these plants and see how that connects in the form of a phylogenetic tree. Have fun drawing all those different features, and I'll see y'all next week.